Good afternoon, everyone, and welcome to the California Immunization Coalition Education Hour. All attendees will be in listen-only mode. If you need technical assistance, please use the chat box on the bottom right of your screen. A link has been sent out in the chat box to all participants that can help troubleshoot any technical issues. During the webinar, you're welcome to ask questions, and you can do so by typing them into the question or chat box on the lower right of your screen. Questions will be answered at the end of the presentation. If we run out of time, the remaining questions will be answered over email. Now introducing your host and moderator, Meg Dorsey.
Greetings, everyone, and welcome. I'm Meg Dorsey, a CIC board member and chairperson for the CIC Education Committee, and I'll be your moderator for today's call. The California Immunization Coalition hosts education hours throughout the year on immunization-related topics to help our colleagues stay up to date on important news and information. Today, we have a very exciting and timely program for you, which is titled New Vaccines, From Development to Clinical Trials and Safety Monitoring. A recording of the slides from today's webinar will be available on the new CIC YouTube channel that you can access via the CIC website, which is immunizeca.org. If you're unfamiliar with this new feature on the CIC website, I encourage you to take a look at it. It's pretty cool. Before we begin our program today, we will hear briefly from Edgar Ednicott on a terrific new media campaign to support immunizations. Edgar is with the California Department of Health Immunization Branch, where he is the chief of the Information and Education section. So Edgar, please go ahead.
Thank you very much, Meg. So I wanted to take about 5 minutes just to give an overview of the Don't Wait, Vaccinate campaign, which hopefully you've heard about from some of our partners here. So if you go to the next slide, Cameron. Perfect, and go to the next one there. Perfect, thank you. So I wanted to show why this campaign was created, and it's really an effort to address the problem that you see here on this graph. We have this dramatic drop in immunization rates among not just children, but adults during the pandemic. And you can see that big decrease of immunization for children in April compared to last year, meaning lots of kids need to get caught up. And we're really interested in seeing how the state does, especially against that typical spike of immunizations that we get in August for back to school. So our goal is really to fill that gap and then some to reach baseline coverage again. We go to the next slide. Perfect. So lots of partners on board. It's a long and growing list, including California AAP, California School Nurses Organization, and several health plans as well. Next slide. Perfect. So we're envisioning a 7 to 8 month campaign, starting with back to school during the summer months, followed by parents of 0 to 2-year-olds, pregnant women. We'll have messaging for adults. And then during the fall and through the winter, seasonal influenza vaccine messaging as well. Next slide. Right, and so the goal here is to get people of all ages to get vaccinated, including adults, pregnant women, and in the fall to get that flu shot. Next slide. So we worked really closely with our partners in developing a comprehensive toolkit, which I hope you've seen. And there's lots of different tools, including talking points, scripts for provider letters, robocall scripts, sample text messages. And we also have a library of social media messages with dozens and dozens of messages that you can tailor and post along with a social media calendar to assist people who use the kit to follow other state partners to put out similar messages each month. So every month we have a different theme, and the theme for July is back to school. So we go on to the next slide. So who is the toolkit for? It's for health plans, providers, social service providers, schools, local health departments, coalitions, and many other stakeholders. Next slide. So, and the campaign is distilled into 3 main messages, and that's don't wait, vaccinate, it's safe to come in, and call your doctor. Next slide. And there it is in its hashtag form. Next. And so I wanted to just, we have 5 different themes that I'm going to show you really quickly. We thought it's really important to acknowledge people's concerns with coming into doctors' offices. So we list out some of the ways that providers are keeping patients safe. The second image there, we want to play on providers as heroes, especially since there are lots of positive sentiments towards first responders these days. Next slide. We offer messages in English and in Spanish. We'll have many more languages as well coming up. Next slide. Here's a sample post for parents 0 to 2-year-olds. I mean, we're gonna have a variety of posts. Some are gonna be interactive, like on the left one, the one on the left, where we ask questions of our users. Some are gonna be powerful quotes, like in the middle. And on the right, some will promote the VSD program, especially during these times when many lost their jobs. Next slide. For the campaign, we did survey parents of 0 to 2-year-olds, and they wanted more pictures of babies. So we did that. Next slide. And then we also have school messages. We understand that there's lots of uncertainty of how schools are going to look like this year, along with immunization requirements. So we created a variety of messaging that can be tailored to whatever is happening in your school district. Next slide. And here's an example. One of those instances will be social, or will be distance learning as a scenario for many schools across the state. So here we say, schools may look very different this fall, but getting your kids vaccinated is still important to protect them against serious diseases during and after the pandemic. Next slide. We have posts for pregnant women. Go on the next slide. We have posts for adults as well. And then the next slide. And then here's the campaign website. You can see the 5 different themes here are broken down on the page. You just click on one of those and you'll see a mix of different messaging. And then we're going to add a 6th theme soon, which will be around promoting seasonal influenza vaccination. Next slide. And then here's some posts from other organizations. We have providers who are using our toolkit and posting. We have local health departments and other partners, schools. LA Unified posted one of our messages as well. So we encourage you to do the same. Next slide. And last slide. So, again, if you haven't seen the website, please go ahead and do that. And then if you have a personal or if your organization has a social media presence, please pick your favorite message and start posting. And again, we have a social media calendar, which is posted on the website that you are welcome to follow. We're hoping that all of these messages can be posted around the same time to help amplify our messaging. So I'll stop there. Thank you.
Thank you, Edgar. What a great new resource. I'm sure we'll all enjoy using that. For our program today, we are so fortunate and grateful to have Dr. Nicola Klein, MD, PhD, and Director of the Kaiser Permanente Vaccine Study Group. She received both her medical degree and her PhD in biochemistry at NYU School of Medicine. Her research has focused on vaccine safety and effectiveness, genetics of vaccine responses, vaccine safety among special populations, and the epidemiology of vaccine-preventable diseases. A more complete and most impressive bio can be found on the webinar registration notice. At the end of today's presentation, our participants will be able to, 1, describe the vaccine development process, 2, detail the importance of safety monitoring, 3, discuss this process in terms of COVID-19 vaccine development. And now we will begin our presentation. Dr. Klein, thank you so much for being with us. The mic is yours.
Great, thank you very much. And thank you very much for the invitation. I'm really looking forward to today's hearing from people today. Next slide, please. Here is just a number of my disclosures, including research support from Pfizer for an upcoming COVID-19 vaccine trial, and then research support from a number of vaccine manufacturers for unrelated studies. Next slide. So today, as we just heard, the goals for today are I will explain the clinical trial phases of development, briefly review the current state of COVID-19 vaccine development, and then describe the U.S. vaccine safety monitoring system. Next slide. So I think we've all been hearing a lot about vaccines and vaccine trials and different phases, and I just want to go through briefly what that really means. So vaccine pre-licensure studies, what does it mean when when vaccines are in pre-licensure or preclinical? It means the same thing. And that means that vaccines are in the laboratory being developed or being undergoing animal studies, as you can see from the little picture. Now, when it gets to human studies or clinical trials, that's when it's called clinical, they're under clinical studies or clinical trials. And there's typically 3 phases of clinical trials. The first one being phase 1, which is considered gross toxicity. And could you go back, please? Yeah. Usually the numbers I put here at 10. It's in the range of double digits. Phase 2 typically is a dosing range and reactogenicity studies, and those are typically in the hundreds amount of clinical trials participants who are recruited. And then there's the phase 3 studies, and typically those are recruiting 1,000 to 10,000 or more individuals, and that what we consider an efficacy study, or sometimes they're actually powered in vaccines for safety studies, and that was the case, for example, the rotavirus vaccine studies, which are powered for safety, and they recruited approximately 70,000 infants. Next slide, please. Now, when you think about clinical trials, you think about randomized clinical trials as being the gold standard of trials, and that's true for many reasons, but that has to do with the fact that you are randomizing individuals. It's a blinded, placebo-controlled design, and you have very close, very detailed follow-up of all subjects who are enrolled in studies. Next slide. However, there are disadvantages of the clinical trial, and that when you have rare or delayed reactions, they can be difficult to detect even with 10,000 or more individuals in a study, because by nature, rare means that there's going to be much rarer than 1 in, for example, 10,000. And further, clinical trials, they typically don't recruit special populations such as individuals with severe underlying health conditions. Often they don't recruit pregnant women. There are just a number of different factors that make someone ineligible for being for participating in a clinical trial. So what that means is you cannot study these populations. And this is particularly of note for the COVID vaccine trials, which are actually trying to address that. And I've actually been told by the FDA in terms of the manufacturers to try and target to recruit disadvantaged populations. But that is typically not always the case and not always done in clinical trials. So that would be really a disadvantage of detailed clinical trial, randomized clinical trials. Next slide. So in this graph, in this cartoon, which came out, as you can see from the reference and earlier in the spring of New England Journal, it was really a paradigm of what is the difference in traditional vaccine development and developing a vaccine during a pandemic. And on the top, you can see the traditional paradigm, which really goes through the phases I just described. And this process can usually takes many years, even up to 8 to 10 years from developing a vaccine to all the way through to the end of licensure and large-scale manufacturing. And you can see that the phase 1s, 2s, and 3s essentially mirror what I just described in terms of how the clinical trials are being done. Now, on the bottom, you can see the outbreak paradigm, which is, thank you, overlapping phases which shorten the development time. And what this means is how this is being done is that the clinical development and all of these decisions are being there's a lot of overlap, whereas on the top portion, you can see there's not as much overlap. On the bottom, you see there's a lot of overlapping phases that are happening, or stages of the development that are happening at the same time. And importantly, the bottom, what you can see here is the manufacturing and scale up is actually starting very, very early in the process. And that's meaning there's going to be commitment being made on a much earlier time frame to large scale manufacturing. And that's sort of the paradigm what's being done at the current moment. Next slide. Now, in terms of what is the current landscape of the COVID-19 vaccines, and this is current, I believe, as of yesterday. If you keep this and look at this in 2 days, it will probably be out of date. But right now, there are at least 140 candidate vaccines in preclinical evaluation. That is, either in the lab or in animals. And I've highlighted, if you can see here, I've starred a few of them because those are the ones that are in the main participating in what's called Operation Warp Speed, which I'll mention in a minute. And that's the US government program to get a vaccine faster. But Merck, for example, that's been targeted, it is developing 2 vaccines. It's not yet in humans, but one of them, they're both based on, one is based on a measles vaccine, and another one is based on their current Merck Ebola vaccine and vesicular stomatitis virus. But there are 27 candidates in clinical evaluation or in humans. And you can see here, there's 6 different vaccine platforms. And what as you can see, there's a wide variety of different sorts of platforms that are being really developed. Some of these you've heard of, more like viral vectors or proteins, some of an inactivated vaccines, and some of them which are newer technologies such as mRNA and DNA technologies for vaccines. So I've highlighted those that are under Operation Warp Speed, and I've taken a little bit of liberty. Pfizer, I don't think is technically in Operation Warp Speed, but they have received almost $2 billion commitment from the U.S. government, and so I've sort of lumped them in that same category. Plus, they've also right up there with Moderna starting a new, they're right there, really pretty far ahead of the pack. Getting their trials going. So I've sort of highlighted those. But what the Operation Warp Speed is, it aims to deliver 300 million doses of a safe, effective vaccine for COVID-19 by January 2021, and as that's part of a broader strategy to accelerate the development, manufacturing, and distribution of the COVID-19 vaccines, therapeutics, and diagnostics. There's not just vaccines. But you can see that there's a lot of candidates, and they may, I would expect that a number of these would probably end up being successful, and I think they're all quite promising, some more so than others, but I think there's a lot of promising candidates you can see from this large list already. Next slide, please. So what is the current clinical trial status of the Operation Warp Speed vaccines? So you can see here there's 4 of the vaccine platforms I've highlighted as part of these Operation Warp Speed. And again, I've sort of taken liberties with the Pfizer and Merck. They've both gotten substantial funding, even if they're not under the exact umbrella. But I sort of consider them very similarly. And I'm going to just focus for a minute on the mRNA ones, the Moderna and the Pfizer, both because although neither one of them are a licensed platform, and that means there's no mRNA vaccine that's currently licensed, but both of these vaccines started phase 3 trials just this week, Moderna on Monday and Pfizer yesterday. I also want to mention that the non-replicating viral vector of the University of Oxford or AstraZeneca, which is also in phase 3, and that is based on a chimpanzee adenovirus vaccine, which is expressing, and all of these pretty much express the spike protein in some form of the coronavirus. The other vaccines are in various stages. As you can see, the Janssen is supposed to start sometime this late July. I haven't heard the exact date, whether that's actually starting or soon to be started, still in July. Novavax has just completed the phase 1 and waiting results. And then Merck, as I mentioned, is not yet in the individuals. The other thing I do want to point out, though, is that in terms of the license platform, you can see that a number of these have not actually been licensed before. The adenovirus for Janssen, that has been licensed, but only in the EU, and that was just a few weeks ago, and that's for the Ebola virus. They also have Ebola virus vaccine, but some of the other platforms do not, so that's just something to keep in mind. So next slide, please. So at this point, I would like to just to stop for a moment and take a poll amongst all the individuals on this call and ask if you could please click on the answer of, are you concerned or have you heard of others who are concerned about the safety of coronavirus vaccines? And the answers are, I and others are concerned. I am not concerned myself, but I have heard that there are others who are concerned and that I am not concerned. And I think we're going to go to the poll, so if everyone could click on their answer.
We'll close the poll in about 3 seconds. We've got about 70% have voted. Okay, I'm going to close the poll now. And here are the results.
Okay, very good. And so I think that this is actually very informative, this very non-scientific poll amongst a large group of individuals who are clearly very interested in vaccines, and I would hazard a guess most of you are vaccine advocates, but that's interesting. So almost 60% of you has said either I or others are concerned. And then the other 35% said I'm not concerned, but that you heard others are concerned. So I think that to show only 7% have no concerns really highlights, I think that probably partially what we've heard is this Operation Warp Speed is what does this mean? Does this mean that it's not going to be safe if it's so fast? And what are we going to do to ensure the safety of vaccines? Now, of course, the clinical trials are going to do detailed, careful, safety monitoring, but I'm really going to spend some time focusing on actually after a vaccine is licensed, how do we monitor the safety of that? So if you can go to the next slide, please. So I just want to think it's worth taking a step back and just remembering that concerns about vaccine safety go back many hundreds of years. You can see here this cartoon from 1802 that there is the smallpox and anti-vaccine society. So you can see that this is a long-standing tradition in this country about being concerned about vaccines. Next slide. So I think we all are very aware that there's also been false scares that from the UK in 1998 when there was allegations of a possible link between MMR and autism. There was immediate fall in rates of MMR. In Ireland in 2000, for example, there were more than 1,600 measles cases. And of course, there's no confirmation of link, and the results have been discredited. And of course, we've seen that result as well here when we had our measles outbreak in this country. Well, we've had several in California, but last year we had a very large measles outbreak in the US, mainly focused on and related to under vaccination of MMR. Next slide. However, there have been real vaccine safety incidents. For example, it's really quite a famous one called the Qatar, the inactivated poliovirus vaccine incident. That was back in 1955, and that was related to because the poliovirus that was during the manufacturing of the vaccine, the poliovirus was not sufficiently killed in several vaccine lots. And so there were some vaccine recipients who were actually infected with live poliovirus. And what that resulted in was 149 cases amongst vaccinated individuals and 6 deaths. And of course, that really shook the public confidence. Next slide, please. And a little more recent, and maybe more relevant to the coronavirus vaccines, there was influenza vaccine and Guillain-Barré syndrome. And that was in February 1976. There was an outbreak of the swine influenza or swine flu. And there was a mass vaccination campaign where 40 million people were vaccinated between October and December 1976. And there was over 500 cases of GBS reported, and there were 25 deaths. And that resulted in the vaccination program being suspended at the time. So I think that there's a lot of history for being concerned about vaccines, but I think there's a lot of, since this time, there's been a lot of progress that has made in vaccine safety. And I think there's a whole science of vaccine safety. And I'll sort of talk about that a little more. Next slide, please. So you could just scroll through for all of the. So when you start to think about vaccines versus drug safety, what makes a vaccine different than a drug? So in general, there's a higher standard of safety for a vaccine. Then that really relates because vaccinees are generally healthy. They may be very young, but not always, but they're just generally healthy at the time they're vaccinated. There may be times when, in fact, often when vaccinations may be universally recommended or mandated. And so we have a much lower risk tolerance for any sort of adverse events. So risk for vaccine adverse events, usually, we want them to be less than 1 in 100,000 or in that range. And whereas when you have a risk for a drug adverse event, depending on what the drug is being used for, some chemotherapies, maybe everybody gets a adverse event, but there's a risk benefit for that versus some, for example, maybe 1 to 1,000, depending on what the drug is used for, what the situations are. But there's a much lower risk tolerance for vaccines when you compare that to drugs. Next slide. So this is a very quite famous graph that came from Bob Chen back. He published it in the mid 90s. And what this shows is that you see on the top, the solid line indicates disease and the dashed line indicates vaccine coverage. And what you can see here in number one, pre-vaccine, when you are in a before there's a vaccine and there's lots of disease incidents, that when there's a new vaccine, there's increasing coverage and decreasing disease, and those until those two lines cross. And at some point there can be some adverse events that may be temporally related to vaccinations. Whether or not they're causal is another question, but they may be considered to be associated, or in the minds of individuals considered to be associated. And eventually there could be a loss of confidence, and that could lead to an outbreak, and then hopefully a resumption of confidence, and then eventually an ideal word, eradication. Next slide. Now, typically, that's supposed to be around the outbreak. That got a little off center, but that circle is meant to be around outbreak. And when I show this slide, this is typically where I always focus my attention. I like to focus the audience attention on. This is where we are. We have a loss of confidence. We might have an outbreak, and we're working to regain confidence to increase coverage again. However, next slide. Next slide, please. Yes. So, actually, can you go back a slide? So, however, in this right now, in this particular situation, and this public health emergency, we're actually in the slightly unique position right now, is that we have of COVID-19, we are actually back at one. We're at the pre-vaccine era, and we are all working very hard to eliminate that and decrease disease, and decrease that solid line, and hope for increasing vaccine coverage. However, next slide. We have to be very aware and thoughtful about making sure that we don't get to the point where we lose the confidence, so that people do receive the vaccine, and then there is no loss of confidence. So I think this is where we really start to think about how to be proactive on really planning for vaccine safety monitoring, and also planning for public communication. Next slide, please. So what is done to ensure the vaccine safety after vaccines are licensed and routinely given in the US? Next slide. There are three main ways. This isn't the only way, but these are the three main ways that vaccine safety monitoring is undertaken in the US. One of the main focuses is from the CDC, Center for Disease Control, Immunization Safety Office, and there's programs for vaccine safety surveillance, as well as research on vaccine safety. Now there's the FDA, which does do some vaccine safety monitoring. It's called the Sentinel Initiative. That is a large monitoring system, which mainly it's insured population of about 100 million individuals in the US. It does focus mainly on drug safety, but it does also do some vaccine safety. And I think that they're planning, as I've just heard recently, they are planning to do some vaccine safety monitoring as part of the Sentinel Initiative. And then finally, the vaccine manufacturers. Now, part of what FDA does is FDA also requires all the manufacturers for newly licensed vaccines to perform what's called a phase 4. I didn't show that in the graph, but that's really called a phase 4 post-marketing safety study. And those are often in the range of 10,000 up to 50 or even 100,000 individuals. And these are individuals who receive the vaccine as part of routine clinical care. These are not pre-selected. These are not enrolled patients. They're just observed and what happens in terms of safety outcomes. And actually, within Kaiser, we have done actually many of these types of studies using our databases within Kaiser. Next slide. So in terms of the, talking a little bit more about what the CDC does and the Immunization Safety Office, there's three main areas in which they focus on. They have the Vaccine Adverse Events Reporting System, which probably a number of you are familiar with, known as VAERS. There's also, they run the Clinical Immunization Safety Assessment Network, and that's known as CISA. And then finally, there's a Vaccine Safety Datalink project called the VSD. And I'll talk about each of these a little more detail. Next slide, please. So VAERS, this is actually an FDA and CDC collaboration that's been around since 1990. And what this is a passive surveillance system. And they collect reports of suspected vaccine adverse events from physicians, patients, really anyone actually can report. Sometimes it's lawyers, oftentimes it is actually lawyers, but many people can submit whatever they think, and there's no assessment of causality. It doesn't have to be due to, but this is if there's a concern or for some reason people can just submit this passively. There's a web-based reporting, and that there is public access to the data. Next slide. And this is actually just an example of, this came just from a few days ago when I went to the website. This is when you go to the VAERS website and you actually look to see report. This is actually what you can do to do reporting. Anyone can go here and put in, the information that's required to submit a VAERS report. Next slide. No, so some of the strengths of VAERS is that it's national in scope. It can be very timely. In fact, the first hints of an intussusception after the rotavirus vaccines came up through VAERS and then was subsequently confirmed in other studies, but it actually first came up through the VAERS system, so it can be very timely. It's relatively inexpensive and cost effective, but what it's mainly best for is to generate hypotheses, what we sometimes call signals. It doesn't mean that a signal that comes up is actually causally related to vaccination, or that doesn't mean it's true or biologically plausible, but it just means that that's a possibility, and you can generate hypotheses from these large amounts of reports. Next slide. Now, some of the weaknesses, though, are because it's a passive system that is voluntary, there's certainly underreporting that happens. There can also be biased reporting, as I mentioned, it could be lawyers or based on news events, etc. It's very much a mix of both causal and coincidental events. There's often inadequate report quality, and people will submit the information that they either have or are available or they can submit. There's a lack of denominators, and that you don't know how many the denominators in terms of the overall. It's not population based in that sense, and there's no control group. And then, as I mentioned, it says here generally unable to address causality. I would say this probably should be changed. It is unable to address causality. Next slide. So another program that the CDC runs is called the Clinical Immunization Safety Assessment Network, called CISA. So this was a network established by the CDC in 2001 as a national network of medical research centers with expertise in immunization safety. And the goal of this is a little different. It's to improve the understanding of adverse events following immunization, what we call AEFIs, at the individual patient level. CISA serves as a vaccine safety resource for U.S. healthcare providers with complex vaccine safety questions about a specific patient to assist with immunization decision making. And really, in simple language, that means it will provide clinical consults for individuals with complicated adverse events after vaccination, and the providers don't want advice on what to do with their patients. And CISA will assist the CDC in evaluating emerging vaccine safety issues. Next slide. And the network sites, we are one of the network sites, but you can also see the other 6 sites: Cincinnati Children's Hospital, Columbia University, Duke University, Johns Hopkins, Vanderbilt, and Boston Medical Center are all the participating sites that work very closely with the Immunization Safety Office to perform these activities. Next slide. Now, finally, I just want to mention the Vaccine Safety Data Link, which you may have heard of, what we call to VSD. So VSD is active surveillance, and some of the highlights of VSD include the near real-time vaccine safety monitoring, or RCA. I'll talk a little bit more about that in a little bit. And it is actually a hypothesis testing system. Next slide. So what is the Vaccine Safety Data Link? It's actually a collaboration between the CDC and 8 integrated healthcare delivery systems. And you can see that here on the map, that there's an obvious overrepresentation of Kaiser as the sites. It's not just Kaiser's, but also health partners at Marshfield Clinic in Minnesota and Wisconsin, respectively, as well as Harvard Pilgrims. But it is a lot of Kaiser sites and Kaiser contributions. But it's a system that started in 1991, and altogether, all of these sites together consist of an enrollment of about 9 million individuals, and that is about 3% of the American population. So next slide. Okay, you can go one more, please. So how does the VSD work? So it's actually a very interesting system. It's what's called the distributed data model. And what it is that all of these different individual healthcare organizations who are participating in the VSD, we all take the integrated healthcare data that is available in all of our electronic medical record systems, and all the sites have a very extensive and thorough integrated healthcare medical record system. And we all create standard files, data. We create standard data files and multiple standard data files. And these include, all vaccines, all individuals, all outpatient visits, all inpatient visits, mostly a lot of procedures, and a variety of other sorts of medical records. And however, we all keep our data. Each site keeps its own data at each site. And what you see here is what's called this distributed data model, and in the center computer is what's called the hub, which is really all it is a way station. You can kind of think that as the way station. And what happens is that for any new study that needs to happen, you can have essentially one of the sites will write a program that gets sent to the CDC. The CDC approves it. All the individual sites approve the computer program, and then they send the program out through the hub, and the hub then sends it out to each of the individual sites, and then extracts the relevant data, not the huge data set, but the relevant data for each study that's being done. And then that data is then sent back through the hub, where it lives for a short while, and then gets transited back to either the CDC or another one of the sites if they're leading the study. So that way, the sites do not send millions and millions of records on millions and millions of individuals. There's not a single place that it lives, for example, in the CDC or other place. Everybody keeps their own data, and there's only study-specific information that's shared for each study. And so that's how this distributed data model works. And this came about for a lot of reasons, but this has been the system in place for at least 15 or 20 years. Next slide. And generally, the priorities of the VSD are to evaluate vaccine safety. And that includes newly licensed vaccines, or new recommendations for existing vaccines, or among vaccines for high-risk populations. Particularly, there's a particular focus at the moment on pregnant women. It has been for several years, as well as other groups as well, sometimes immunocompromised, sometimes premature infants, individuals with HIV, for whatever particular group it is. But there has been other targeted studies as well to evaluate vaccine safety. Another priority is to develop new methodologies for vaccine safety assessment, and I will have to say that VSD has really been a leader in this, and actually probably, I would argue, a global leader in creating new epidemiological methods for doing vaccine safety assessments. And then finally, we do test hypotheses generated by signals from VAERS. Also the clinical trials, that can be a good source of trying to, if there's a signal that maybe raises an eyebrow, but is not enough to make it's not statistically significant or something, but just a slight imbalance in the vaccine group over the placebo group, that's something to keep an eye on. We could do a hypothesis for that, or other platforms such as the Sentinel network, for example. They may find a signal in their large, some sort of platform, for example, like that, because we have such detailed, there's such detailed data on our people in the healthcare system, on our databases. We can actually access the charts very rapidly and evaluate to see if those cases, for example, of whatever outcome, let's say we're concerned about Guillain-Barré syndrome, we can rapidly take a look at the medical record to determine that whether those are actually a case of a new case of Guillain-Barré syndrome, or is it someone who had a history of Guillain-Barré, for example, in the distant past, for example. So next slide. So the strengths of VSD are that there's a large sample size. Like I mentioned, about 9 million individuals. The electronic medical record data is available for epidemiological studies, and we can calculate incidence rates and attributable risks because the VSD knows the population, knows actually how many people are in its population. It's relatively quick and timely. And then there's also rapid cycle analysis. And what that is that's something that was developed about 15 years ago. And what that is an ongoing surveillance for multiple vaccines. And this involves when there's a new vaccine that's licensed, we generate data files that are updated on a weekly basis. And the data can actually often be analyzed on a weekly basis to do an early detection of any safety signals that may be coming up. And so this has been a very powerful, well, okay, I'll go back to that. So, but the limitations of VSD is that the VSD members are generally well vaccinated, so they can be difficult to find unbiased controls. Populations are not large enough for some hypotheses. For rare outcomes, 9 million is still not large enough. It can be difficult to study adverse events following immunization that have delayed or an insidious onset. And this is a big challenge, not just in the VSD. This is actually one of the big challenges I would say in the field of vaccine safety, is trying to understand and trying to come up with ways to look for new onset, for example, autoimmune conditions that may take many months, even years before the diagnosis is made, but it takes a long time, and the symptoms can start, months to years earlier for the diagnosis. And that's a big challenge in trying to understand in vaccine safety studies. And then it's also expensive. This sorts of very intense, lots of infrastructure that's constantly required to maintain these data sets and data files in large amounts. It's an expensive project. Next slide. So talking a little bit more about the rapid cycle, which I sort of mentioned, has become a very powerful tool for understanding and looking at vaccine safety. And this, again, it's an ongoing surveillance of multiple vaccines, and it's usually one vaccine at a time, but we do different vaccines at different times. And it's a weekly update of the data. And while that doesn't sound, maybe that doesn't sound too impressive, before the rapid cycle analysis was created, our typical data sets that we do are big epidemiological studies that span 10 years of data. We only update those once a year. So if you want something timely and new, this creation of weekly updates of data was really a very big innovation and step forward. And in fact, I'll give you an example of an RCA study was the increased likelihood of febrile seizure 7 to 10 days after the combined measles, mumps, rubella, and varicella vaccine. Can you go back, please? When compared with separately administered same-day MMR and varicella vaccines. And I'm sure many of you are aware of the increased febrile seizures, but this was actually first detected in the RCA analysis after MMRV was licensed. And next slide, please. So just to touch on that quickly. So febrile seizures are the most common neurological event in childhood, affecting up to 5% of children ages 6 months to 5 years. And at the time of licensure of MMRV, MMR vaccine had been known to be associated with febrile seizures 1 to 2 weeks after vaccination. And in clinical trials, MMRV versus separate same-day MMR and varicella vaccines had higher rates of fever and measles-like rash 5 to 12 days after vaccination. But this is exactly what I was mentioning in the very early part of my talk, that although clinical trials, we did detect this higher rates of fever, they were not large enough to find any difference in febrile seizures, because febrile seizures are much rarer than fever. Next slide. So as part of the RCA study, when we did the weekly analysis of this, looking at seizures after MMRV versus MMR and MMR plus V, MMR plus varicella, this actually came from the paper that we published about 10 years ago now, but the actual data from the weekly preliminary data looked very much like this. And what we found was that you can see that the blue line represents, these are seizures in the emergency room among 12 to 23-month-olds after the first dose of measles-containing vaccine. And you can see the blue line represents MMRV, and you can see that blue line is roughly twice as high as the pink line. The pink line is separate MMR plus varicella, and then below MMR alone. But you can see that that occurred very sharply between days 7 to 10. Next slide. And when we actually did the analysis, what we found was that the risk of febrile seizure 7 to 10 days after MMRV compared with MMR plus varicella from 2000-2008 was roughly twofold higher increased risk. You can see in the box. And what we found was that there's a risk difference, or 4.3 per 10,000 doses. And what that means really is that for every 2,300 doses of MMRV given to 12 to 23 months old, instead of MMR plus V, there was one additional febrile seizure 7 to 10 days after vaccination. So I think this is probably, and this led to the MMWR, in which the CDC recommended that unless parents actually request MMRV, and toddlers should be given separate MMR plus varicella vaccines. Now, this, I'm sure everyone on this call is very familiar with this. This is actually quite old news now, but it really just highlights the power of these rapid cycle analysis, that we rapidly found that there was this risk of febrile seizures, and then we were able to follow up with a formal study, and which we published here, and actually led to changes in vaccine recommendations. So next slide. So there's been, since that wasn't the first one, but it was the first one to detect a signal. But you can see there's a list of other vaccines that have undergone RCA studies. There have been, the PCV13 also found an increase of fever after, after there's a flu with the febrile seizures. That was also seen with PCV13 when there's concomitant. But most of the other RCA studies have not detected any increased risk or increased safety findings. And we are currently planning for a COVID-19 RCA study that will be a very important safety monitoring. Next slide. So this here is, I debated whether to show this. These are all this is very preliminary and under development. They're still working on whether these are all going to happen. But so I thought I just mentioned these are the types of studies that are actually being considered with the CDC. And for the CISA, I mentioned there's a clinical consult in general to do for adverse events for patients. And that's also being planned actually for COVID-19 vaccines for individuals, to actually provide a clinical consult service for adverse events for individuals. And then also, they're also planning to potentially do some observational safety studies after enrolling patients for COVID-19 vaccines amongst pregnant women, adults, and possibly children, but these are still under development. For VSD, there's a number of studies that are under consideration. Of course, I mentioned the rapid cycle analysis, and what this is, and I didn't mention, but these are targeted outcomes. Typically, there's a list of maybe 5 to 10 outcomes that are of high priority to monitor for a particular vaccine. And those are based on either clinical trials or biologic plausibility or other concerns. But there's also a data mining, possibly some data mining vaccine surveillance, and that's really to look for broad outcomes without any pre-specified hypothesis, but to look for any unanticipated outcomes that we hadn't considered. There's going to be there's consideration to do a vaccine safety study in pregnant women and their infants to monitor for vaccine-enhanced COVID-19 disease, to assess the impact of changes on healthcare utilization due to the pandemic on the rates of vaccine adverse event encounters, to monitor for multi-system inflammatory syndrome in children after vaccination, and assessing vaccine safety knowledge, attitudes, and belief associated with acceptance and refusal of COVID-19 vaccination, which I think is going to be quite important given the earlier part that we talked about earlier today. So next slide. So I just want to I'm hoping that with this vaccine safety discussion and really description of what are the safety systems in place and that will be in place for COVID vaccines and will be enhanced as well as what is currently existing. Next slide. We will not see that, have that fear, but actually that the public and that we all agree that the benefits will far outweigh the risk of vaccination. Next slide. And I think with that, have, I think, a little bit of time for vaccine. Excuse me, for questions.
Vaccine questions.
Yes, that's right.
Okay. Thank you very much, Dr. Klein, for a great presentation on a really important and timely topic. I'm sure that we've all benefited greatly from your expertise and experience. This was really interesting. So if our listeners have been waiting patiently to ask a question, now is the time. So please type your question into the question section at the bottom of the screen. You could also place your question in the chat section. And while we give everyone a moment to type in their questions, I would like to remind our participants that following the program, an evaluation will pop up on your screen. And we truly appreciate your responses. The California Immunization Coalition produces at least 3 to 4 education hours per year, and we rely on your feedback for preparing and improving these calls. So please take a moment and give us some feedback. So again, thank you so much, Dr. Klein. We do have several questions in queue here for you. The first one, do you anticipate that once we have a COVID vaccine available to us, it will be licensed based on immunogenicity, efficacy, or both?
So I'm assuming you mean the emergency use authorization, the EUA, and the FDA has been they've published some guidance late last month in which they said that in order for a vaccine to be granted an EUA or even a license, they have to demonstrate at least 50% efficacy in clinical trials, and with the lower bound of 30%, which means that when you do make confidence intervals around the efficacy, they can't range any. It can't be any lower than 30% on the lower bound. So it will be based on efficacy, and of course immunogenicity is very important, especially if there could be ultimately determine a correlate of protection, and so that future studies can just perhaps might be able to use immunogenicity. But right now they have stated that trials must show efficacy of 50%.
Great, thank you. Another question came in about age indication. You did a great job outlining all the various COVID candidates. Are they doing studies on children, or what is the age range expected for licensure of a new COVID vaccine?
Yeah, that's a great question. So right now, all the trials are being done in adults, and all the trials in this country are all being done in adults as 18 and over. And there's some special focus on trying to get looking at older individuals as part of the studies, but they're all in adults right now. You know, I don't know I'm sure that there's plans to do studies in children, but I don't know what they are. And I think that those, I suspect, are going to happen after some sort of conclusion, or at least some preliminary looks within the phase 3 in adults. That is part of why CISA is actually thinking about doing some clinical observational clinical studies in children, but I think that that's again an open question because it's not clear exactly when and how the childhood studies will need to be done, but they will need to be done for sure. That's an important question.
Thank you. Another question, do you anticipate a new coronavirus vaccine to be a one and done, or do you think this might be something where we have a series of vaccinations or revaccinations as we have with flu?
Well, that's also a great question. I will say that right now, most of the vaccines, just to be very clear and literal about that, most of the vaccines being tested right now are actually 2-dose vaccines, so it's going to require at least 2 doses. Whether that's it, or whether it's going to need to be, you know, a boost or an annual, but I think that right now there's so much unknown about coronavirus disease that I think everyone is expecting a surge in the fall with winter respiratory season, but does that mean it's going to come back year after year? Possibly. I don't think we know, but I think right now that's one of the great questions. We don't know how long, if these vaccines work and they're efficacious, how long do they last for? I think that is a completely unknown question right now. I don't think it's really well understood how long protection lasts against natural disease. And so I think these are all great questions that I don't think we really know the answer to right now.
Okay, another question that we have is regarding any thoughts or information that you have on co-administration of a novel coronavirus vaccine and seasonal flu vaccine. For example, a participant asked about possibly vaccinating an asymptomatic COVID patient with a flu vaccine. So any thoughts or comments on co-administration of flu vaccine and COVID vaccine?
Right. So all the clinical trials right now do not allow for co-vaccination, but they do all recommend and encourage flu vaccination, just not together with the coronavirus vaccines that are being tested. And that's fairly typical for clinical trials, that unless they're actually specifically trying to evaluate co-administration, which is usually something that's done later after a little more of the initial phase 3s are done. But I don't know that that's going to happen at all in this case, but I think they're not allowed to, in terms of the clinical trial, to be co-administered for the two vaccines, although they will all be encouraged to, all the individuals will be encouraged to get the flu vaccine, just not at the same time. Regarding the question of giving the flu vaccine to an asymptomatic coronavirus patient, I would have to think about that. I would probably discourage that at the moment, just because I don't think we always know whether someone is asymptomatic or pre-symptomatic for coronavirus if they test positive. So I think it depends on, at what point they're being, they're identified. But I think I would probably at this point be inclined to wait a little bit to give the flu vaccine, but I would have to think about that a little bit more.
So the next question is not really straight out of the research. It's more of your personal take or your personal experience. As we are all aware, COVID is hitting our communities of color much more heftily, and those communities are also much more vaccine hesitant. So do you have any thoughts or suggestions, or how might you phrase a message on encouraging communities of color to receive vaccination?
Well, I think we are going to have to be very honest with our communities, all communities, not just community of colors, but all, in the public, in terms of what we know, what we don't know, and what's realistic to expect from a vaccine, and what I think is unknown at the moment, and to be honest about what we do know, and not, I think, over promising things that we just don't have any information about. That being said, I mean, communities of color, I would say that I think there's variation in the different communities on vaccine hesitancy. I think maybe depending on the community, I don't know that I would say all communities of color are vaccine hesitant, but I think that's many of them are. I agree, and I think I'd probably use the same approach that we've actually started to look at for just our vaccine hesitance populations and our members, and really emphasize that it's a partnership, that we're all trying to protect individuals in the community or in your family, that I think we are in the same, we're all trying to do the same thing, that the individual, we're trying to protect the individual, and the individual wants to protect themselves and protect their families, and that vaccination is going to be the way to protect everyone, and be honest about what we don't do and don't know. That's what I would recommend.
And then I think we have time for just one more question. Question related to vaccinating a person who has already been diagnosed with and recovered from COVID. Do you anticipate that we'll vaccinate those who are already recovered?
I know these are very difficult questions. I think that these are all going to be eventually addressed. I think ACIP is really looking at this very closely in terms of who should be vaccinated, who should be targeted, who's going to be priority. I think in the beginning there's going to be quite a detailed prioritization in terms of vaccination population, because there's going to be limitations, at least for the early stages. Eventually there'll be hopefully not, but I don't know that would be considered a person who's known to have recovered. I'm not sure that they would be considered a high priority group or not. I think that's a good question. I don't think we have any data. I think the only thing the only very minimal information we have about that are so far in the phase 1/2 trials of the individuals who received 2 doses of some of they received on the second dose, that they saw a boost in their immune response after the second dose of vaccine. And that's probably not a great analogy, but that's probably the best we have at the moment in terms of they did see an increasing in their antibody responses.
Well, thank you again so much, Dr. Klein. We're coming up to the end of our hour. Do you have any final thoughts or comments that you'd like to make?
Well, I just want to thank you very much for the opportunity to speak with all of you today, and I think thank you very much.
All right, Dr. Klein,
and
we will make sure to take down the rest of the questions, and we will answer them and send out an email with those questions.
All right, and we hope this past hour has provided you with some useful information to prepare you for what lies ahead with respect to the introduction of a new coronavirus vaccine. We'd also like to thank the members of the CIC Education Committee for support in planning and coordinating the call. And a very big thank you to all of you for spending your hour with us today. Please remember to complete the evaluation form that will automatically pop up after this broadcast and give us your feedback. Thank you again for joining us, and enjoy the rest of your day.